Hello there, welcome to this webinar. My name is Parham Raisi. I'm professor of physiology at Isfahan University of Medical Sciences. During this series of webinars, I want to explain about different methods of extracellular recording, and I have to say one of the important aspects and feature of your recording is interpretation. By new devices, you can achieve a lot of data. At the end of your recording, you have a lot of data that you don't know about the interpretation of them. If you want to have a good record and also you interpret them in the best way, you have to know about basic features of electrical properties in our cells and tissues. By this series of webinars, I want to explain about this basic feature, and continue in next parts of my webinars, I will explain about, for example, electroencephalography in animal model, single unit recording, field potential, and other types of extracellular recording. In this session, first I want to introduce some basic features of electrophysiology, definition of electrophysiology, and different type of electrophysiological recording in human and in animals. First, about the meaning of electrophysiology. What's the meaning of electrophysiology? Electrophysiology is a study of electrical properties of cells and tissue. As you know, all of the cell membranes are polarized. Inside the cell is negative than outside. This polarity achieved by three main reasons: sodium-potassium pump activity, large ions that are confined inside the cell and can't exit the cell, therefore they remain inside the cell and make inside the cell negative. And the third one, that is the most important, are small ions that can pass easily through a cell membrane, and according to selective permeability to these ions, they can produce the main part of membrane potential. During resting membrane situation, the main ion that communicate in production of membrane potential is potassium. As you know, in diffusion, by progress of diffusion, we have equality between two sides of the membrane for that ion, for that molecule. For example, when we have high concentration, low concentration, we have diffusion from high concentration to low, and by time we have equality between two sides. But this situation can achieve only for uncharged ion. For charged ion, same to sodium or potassium ions, by time when we have concentration difference, when sodium move from high concentration to low concentration, by this action we have increased positive ion in this side and decrease in other side. Therefore, by time we have a change in the voltage of two sides of the cell membrane. By this action, we don't achieve equality in equal concentration between two sides, rather than our equilibrium for ions achieved in a voltage. For example, when we have a high concentration in one side and in other side we have very high voltage. For example, if in extracellular fluid we have sodium 10 times than intracellular fluid. However, if we make inside the cell positive enough, about 61 millivolt during this situation. However, chemical gradient wants to send sodium from extracellular to intercellular, but because inside the cell is positive enough, therefore also the positive environment inside the cell want to repel each positive ion same to sodium. Therefore, in this situation, for ion, when we have free diffusion, therefore they achieve or approach to equilibrium between two sides. But for ion, equilibrium achieved for in a voltage difference rather than in equality of the concentration of two ions or molecule between two sides. And because during resting membrane potential, all of the cell membranes are more permeable to potassium, and equilibrium voltage for potassium is minus 94. Therefore, in most of the cell, inside the cell is negative than outside because to selective permeability to potassium. And this amount of voltage is different between different types of cells. For example, for large neuron and skeletal muscle cells is about minus 90. In small neurons is about minus 70, or in skin cells is about minus 20. Therefore, all of the cell inside the cell is negative. But amount of this negativity is different, and polarity different between different types of cells. Why? Because we don't have equal concentration difference between all of the cells. In some cells, concentration difference between inside and outside, for example, is 10 times, sometimes 5 times, or higher or lower. By Nernst equation, we can achieve the exact value of this voltage. And also in all of the cell, we don't have only permeability to potassium, because we have a small permeability to sodium or other ion, same to hydrogen, same to chlorine and calcium. Therefore, each of these ions want to drag the voltage toward their equilibrium voltage. But the ion that is more permeable, the voltage of the cell membrane is nearer to the equilibrium voltage of that ion. And in resting membrane situation, we have a higher permeability to potassium. Therefore, potassium affects membrane potential and produce the most part of this polarity that is negative. And during action potential, because during action potential we have higher permeability for sodium, or in some cells for calcium, therefore this ion drag the voltage toward the equilibrium voltage and it gets more positive. We have two types of electrical propagation along the cell membrane after if you excite a cell. However, all of the cell membranes are polarized. Inside the cell is negative than outside, but some of the cells are excitable. Which cells are excitable? The cells that have enough number of voltage-gated channels by a stimulation of this membrane, because affecting the structure of protein of these gated channels, that these voltage gated channels are open in one voltage and they are closed in another voltage. And therefore, by stimulating, I mean by increase the voltage of the cell membrane, because of the change in the structure of these proteins, they get open, and by opening some positive ions, same to sodium or calcium, rush into the cell and increase the voltage and produce depolarization of membrane. This electrical change by stimulation can propagate along the cell membrane by two ways. We have two types of cell membrane propagation. The first one is gradual change or electrotonic conduction, and another one is all or none principal or action potential. If I want to show you, consider this is a neuron, this is an axon. If you stimulate inside the cell, at first inside the cell is negative. Excuse me, by pain is difficult for. Inside the cell is negative and outside is positive. Therefore, we consider inside the cell negative and outside the cell positive. However, at first I have to say, electrophysiologists have agreement that consider the voltage of extracellular fluid equal to 0, and we attribute all of the voltage difference between intracellular and extracellular, and we attribute all of them to intracellular. For example, sometimes we say that the voltage of this cell, the polarization of this cell or resting membrane potential is equal to minus 90. Maybe inside the cell is minus 70. In extracellular cell fluid is minus 70, positive 20. You can see inside the cell is negative than outside, about minus 90. Because this is difficult, algebraical difference is minus 90, but it's difficult for us to calculate the exact voltage during calculation of it. Therefore, we consider the extracellular voltage equal to 0, and we attribute all of this voltage to intracellular. Anyway, inside the cell is negative. If you stimulate a cell and you open a channel, for example, by neurotransmitter through ligand-gated channels, or by physical stimulation through mechanical gated channels, sodium ions, because inside the cell is negative, therefore positive ions rush into the cell and make this part of the cell positive. This is resting membrane potential, for example, that is minus 19, and also this is a threshold level, that is, for example, minus 90. If you stimulate a neuron and open some ligand or mechanical gated channels, inside the cell get positive and increase the voltage, but this voltage don't reach to threshold level during this situation, because we don't have any change in the structure of voltage gated channels. We don't have any opening of extra channels by through ligand gated channels, and we don't have any action potential, and this voltage come back to previous level. However, inside the cell get positive and then other part of axon. Therefore, these positive ions diffuse toward the place that we have higher number of negative ions. And by production, a small circuit, because as you know, we have some leak channels that potassium positive charge potassium exit, and this part get more positive, and when this positive ion come into the cell, in this part get negative, and this potassium exit the cell and make in this part positive, and therefore this positive ion move to this negative part, and therefore they produce complete circuit. And by this small complete circuit, this current can move along the cell membrane. However, this type of electrical propagation or electrotonic conduction has two important features. The first one is decreasing manner because of these potassium leak channels and also because of activity of sodium potassium pump. By propagation and by time, this voltage decrease because these positive ion escape from the cell, and therefore we achieve the previous equilibrium that achieved, for example, in -90 or in resting membrane potential. Therefore, by a Lenz's constant, this positive stimulation decrease during electrotonic conduction. Also, in electrotonic conduction, in spite of action potential, we have graded response. In action potential, if you reach to threshold level, you achieve all of your response, and if you stimulate more than threshold, also you achieve only this top of action potential. If you don't reach to threshold level, you don't have any action potential. Without reaching to threshold level, we don't have any action potential known. If we achieve or we near to threshold level, we achieve all of our response, all or nothing principle. But for electrotonic conduction, we have graded response. If you stimulate, insert a small stimulation, you have a small EPSP. If you stimulate and insert more positive ion, you achieve a higher or larger EPSP. Therefore, we have two manners. Decreasing manner, by a Lenz's constant, it decrease until it reach to 0, and this appear from a neuron. And also it's graded. When you have a stronger stimulation, you achieve a stronger response. Also, I have to say, action potential can transmit only positive stimulation. Sometimes through inhibition, you have hyperpolarization. Also, hyperpolarization can propagate along the cell membrane by electrotonic conduction.